Hi everyone. This module is on revision. Okay, revision with the MCQ series. So hope you have given the test. Hope you have given the test which I have posted earlier. So if not, please go back, give the test, and then come back for this discussion video. Okay. So the first question is, all of the following are taking place inside the mitochondria except. So citric acid cycle, yes, it takes place in the mitochondria. Electron transport chain, of course, it takes place in mitochondria. Beta oxidation of fatty acid, all these three pathways take place in the mitochondria. So de novo synthesis of fatty acid, it takes place in the cytoplasm and not mitochondria. So this is the answer. Now, all are true about ionophores except. What are ionophores actually? Ionophores are nothing but they are substances that reversibly bind the ions. Okay, so these are substances that will bind ions. So they bind ions and they transport the ions across the membranes. Okay, and they are used for transporting antibiotics. So mainly what are these ionophores? These ionophores can be antibiotics, right? And example is, we have examples, but before that you have no types of ionophores. We've got channel forming ionophores. Yes, channel forming ionophores and mobile carriers. So example for channel forming ionophores is gramicidin. Okay, gramicidin. And example for mobile carrier ionophore is valinomycin. Valinomycin. So now let's look into the options. Whether it transport antibiotics, yes, it transports antibiotics. Example is valinomycin and gramicidin. Yes, examples are this. And valinomycin is transported by channel forming ionophores. No. So what is this channel forming ionophores? Consider this is the cell membrane. It forms a channel through which the ionophore pass inside the cell. Or there are mobile carriers wherein it will bind to this and these carriers will transport it inside the cell. So this valinomycin is transported by mobile carrier ionophores. Okay, and they transport ions across membranes. Okay, yes, it transports ion across membranes. So the option C is the answer. Now, DNA and histone proteins form a network of thread-like structures called chromatin. Yes, the lengthy DNA is compacted into a tight structure or a shortened structure by histone proteins. Yes, by the histone proteins binding to these DNA and folding them multiple times. They form thread-like structures called chromatin. And protein biosynthesis initiates at ribosomes. Initiates at ribosomes. Yes or no? In where does this ribosomes present? What is endoplasmic reticulum types? Rough endoplasmic reticulum and smooth endoplasmic reticulum. In the rough endoplasmic reticulum is rough because of the presence of ribosomes. So whenever there is the protein synthesis is actively happening, these proteins ribosomes will be actively synthesizing these proteins, and that will later on enter endoplasmic reticulum wherein your post-translational processing will take place, and then which will enter into the Golgi apparatus, where in the Golgi apparatus protein sorting, packaging will all happen, and it will be distributed to whichever area these proteins are supposed to go. So ribosomes is the answer here. Protein biosynthesis initiates at ribosomes. And protein sorting and packaging takes place as Golgi complex. Yes, it is Golgi complex. And next, all are true about peroxisomes except. All are true about peroxisomes except. Let's read the options first. Catalase converts hydrogen peroxide to water and oxygen. Peroxisomes are used in oxidation of short-chain fatty acids. Peroxisomes help in synthesis of plasmalogens. Peroxisomes generate hydrogen peroxide in the process of removing hydrogen from organic molecules. So let's see, in peroxisomes, what are the two enzymes? The two enzymes are catalase and peroxidase. What does catalase do? Catalase converts hydrogen peroxide to water and oxygen. That is the function of catalase. Peroxidase, it's involved in lipid peroxidation. Okay, so from lipid peroxidation of or if any organic molecules, hydrogen has to be removed, it combines with oxygen to form hydrogen peroxide. So peroxidase itself involved in lipid peroxidation, it generates free radicals. Okay, so this generating free radical is the right answer, right option. And catalase converts hydrogen peroxide to water and oxygen, it is right. Peroxisomes are involved in synthesis of plasmalogen, that is also right. But fatty acid oxidation, where does fatty acid oxidation take place? It takes place at two areas. One is mitochondria, right? Most of it takes place in mitochondria. And the other one is peroxisomes, okay, peroxisomes. Very long chain fatty acids do not have any, do not get oxidized in the mitochondria. So they have to enter the peroxisomes for oxidation. Okay, so peroxisomes are used in oxidation of short chain fatty acid. It is not right. Short chain fatty acids are oxidized in mitochondria. So very long chain fatty acid is right. Okay, so this option B is the answer here. So next question is membrane junction complexes are all except. Membrane junction complexes, desmosomes, tight junctions and gap junctions. These three are membrane junction complexes, right? They provide links between two cells, two adjacent cells. So they provide communication pathways, or they provide a connection between two cells, right? So that the desmosomes, tight junctions or gap junctions are the membrane junction complexes. They help in connecting two cells, they help in stabilizing the cells in its structure, and also they help in communication between cells, or if any molecules have to be transported between the cells, that also happen through these membrane junction complexes. Whereas what is a receptor? Receptor is a transmembrane protein or an integral protein in the plasma membrane. Okay, transmembrane protein. So to these receptors the ligands bind and further function of these ligands will take place. Okay, so these receptors will be transmembrane protein or integral membrane protein. These receptors to which ligands can bind. So the answer for this is receptors. Next, ion channels or examples of. Ion channels or examples of. So before seeing this, let's see the classification. So transport mechanisms, how do you classify? You classify transport mechanisms into active transport and passive transport. Okay, so active transport, you again divide them into primary active transport and secondary active transport, and passive transport you divide them into simple diffusion, okay, diffusion and facilitated diffusion. So facilitated diffusion is again classified into carrier mediated, the help of transporters or carrier proteins, and then channel proteins. Okay, channel proteins can again be divided into ion channels, it can be ionophores, yes, or can be aquaporins, water channels. Yes, so this is the overall classification of the transport mechanisms. Now, ion channels are now examples of what? They are examples of facilitated diffusion. They are selective transport. So, only one particular ion can, selective transport of ions. So, one particular ion can pass through the ion channels. So, what are the types of ion channels? They are ligand gated, or they can be voltage gated or mechanical. Yes, voltage gated or mechanical. So the ion channels are examples of facilitated diffusion. Now all are true about the above image except. So what is this image? This image is nothing but sodium potassium ATPase pump. So sodium potassium ATPase pump. So what is the sodium potassium ATPase pump? It is a example of active transport. Okay, what active transport? Primary active transport. It's an example of primary active transport. So what is its a function? The sodium, here three sodium ions are pumped out inside this from inside to outside the cell. Okay, and two potassium ions are pumped out from outside to inside the cell. So this is the function of the sodium potassium ATPase pump. So sodium is, yes, so from inside the cell, this is outside the cell. So three sodium ions are pumped out from inside to outside the cell, and two potassium ions are pumped out from outside to inside the cell. For this, what do you need? You need ATP. What do you need? You need ATP. Yes, so ATP driven sodium potassium pump is nothing but a type of primary active transport. A primary active transport is nothing but wherein the substances are transported from an area of lower concentration to an area of higher concentration. This is against electrochemical gradient. Yes, it is against electrochemical gradient. So you are actually pumping something from an area of lower concentration to an area where the ions are more there. So higher concentration. So against electrochemical gradient you are pumping this. So it needs ATP. So it needs energy. Okay. So the sodium potassium ATPase is one example. Also potassium hydrogen ATPase pump in gastric mucosa. That is also another example of this. Understood? So let's see the options here. This is an example of active transport. Yes, it is an example. It takes place against electrochemical gradient. Yes. It requires ATP. Yes. But the binding site for sodium is on the extracellular surface of the cell. Do you think sodium has to be pumped from inside the cell to outside the cell? Yes, from inside the cell to outside the cell. So binding site for sodium will be present inside the cell on the intracellular surface only. Yes, so this is the answer for this question. This is wrong. It should be present in the intracellular surface of the cell. So next question is unipotent is match the following. Okay, unipotent, sympotent and antipotent. What is unipotent? Unipotent is where one solute passes in one direction. Okay, and sympotent is nothing but two solutes pass in the same direction, or you can also call it co-transporter. And antipotent is two solutes, two molecules pass in the opposite direction. Okay, so unipotent, what can be an example? Facilitated diffusion of glucose. So consider this is the GLUT transporter. GLUT transporter. Glucose is taken up inside the cell. This is a facilitated diffusion of glucose. It is carrier mediated. The transporter here is the GLUT transporter, glucose transporter. So unipotent is a the example for unipotent is facilitated diffusion of glucose using GLUT transporters. Yes. And then sympotent. Sympotent, what can be an example? Sodium glucose transporter. Yes, along the apical side of the apical side of the intestinal membrane, intestinal cell, the sodium is taken inside the cell, intestinal cell along with glucose. Okay, from the lumen inside the cell, sodium is taken in along with the glucose. So this is sodium glucose transporter. This is a sympotent. Yes, so sympotent is sodium glucose transporter, and antipotent is sodium potassium. We've already seen sodium is pumped out and potassium is pumped inside the cell. So here this moves in the opposite direction. So antipolar example is sodium potassium ATPase pump. So the process by which cell engulfs bacteria and debris from damaged tissue is called. So you all know the answer for this. This is phagocytosis. Yes, the answer is phagocytosis. So how can we classify vesicular transport? How can we classify vesicular transport? One is endocytosis. Yes, the other one is exocytosis. Endocytosis and exocytosis. Yes, so endocytosis is again classified into pinocytosis and phagocytosis. Okay, pinocytosis and phagocytosis. Pinocytosis is again divided into two, which is fluid phase pinocytosis and receptor mediated pinocytosis. Understood? So here exocytosis, what is exocytosis? Here, so exocytosis is nothing but from inside the cell, any substance which has to be externalized, it forms vesicle. Yes, it forms vesicle. From inside the cell, it forms vesicle. It refuses with the plasma membrane. Yes, it fuses with the plasma membrane. It opens up and then the contents are externalized. Okay, the contents are externalized. The example for this exocytosis can be hormone insulin release. Okay, insulin release from beta cells of pancreas or acetylcholine release from the presynaptic nerve terminals. Presynaptic nerve terminals. Okay, these can be examples of exocytosis. Endocytosis is pinocytosis and phagocytosis. What is pinocytosis? It is nothing but cell drinking. Okay, so some amount of extracellular fluid is constantly being taken up inside the cell. So what will happen? It will form one vesicle-like opening in the plasma membrane wherein the contents are taken in and it is transported inside the cell. So here, if it is fluid phase, it is non-selective. Okay, if it is receptor mediated, it means after the bind. If it is receptor mediated, let me tell you. If a receptor, for example, LDL molecule, we have an LDL receptor. Okay, whenever LDL molecule binds to the LDL receptor, it forms clathrin coated pits. Okay, finally what happens, this LDL is then absorbed inside by forming a vesicle into the cell. Okay, so that's how LDL is transported into the cell. So by binding to a receptor, after which the receptor will be recycled back to the plasma membrane. So this is an example of receptor mediated endocytosis. And what is phagocytosis now? Phagocytosis is nothing but it is the engulfment. All the bacteria and debris are being engulfed inside the cell or phagocytosed inside the cell, and it can later fuse with lysosome wherein this bacteria will be digested. Okay, so this is what is phagocytosis. So the process by which cell engulfs bacteria and debris from the damaged tissue is called phagocytosis. So all of the following lipids are components of biological membrane except. Yes, in the biological membrane, it's a lipid bilayer, we all know, right? So lipid bilayer is made of mostly phospholipids. It's made of mostly phospholipids. Okay, also you have cholesterol in between. Also you have cholesterol in between. So how are these phospholipids arranged? It has hydrophilic head on the outer side and hydrophobic tail on the inner side. So this is how the lipid bilayer is arranged. Okay, and also on the outer surface of the cell membrane, some oligosaccharide units will get attached to the lipids here. So this is nothing but glycolipids. Okay, from some oligosaccharide sugar units can bind to these lipids on the surface. So can form glycolipids. Also cholesterol is embedded in between in the plasma membrane. So cholesterol is also present. So what is not present? Triacylglycerol is not present in the cell. So the answer is C, triacylglycerol, which is not a component of biological membranes. Now, what are all are true regarding transport mechanisms except. So, whenever you get a MCQ with long options, lengthy options, please don't panic. Just read and see. It will be easy for you. Okay. So, all are true regarding transport mechanisms except. So, we have to find out which is false. So let's read option A. Sodium dependent transport of ions and nutrients along their electrochemical gradient is secondary active transport. So what do we know about secondary active transport? Secondary active transport, it indirectly utilizes ATP. Primary active transport, it directly utilizes ATP. Secondary active transport indirectly uses ATP. Okay, so let me tell you a sodium glucose transporter. Sodium glucose transporter, which is present on the apical side. Sodium glucose transporter. So sodium along with glucose is transported inside the cell. Okay, this directly does not require ATP, but sodium if it has to enter inside the cell, sodium if it has to enter inside the cell, the sodium concentration inside the cell should be maintained by what? Sodium potassium ATPase. Okay, it should be maintained by sodium potassium ATPase pump. So this requires ATP. Okay, so the sodium concentration is very crucial for the sodium glucose transporter to function. So in turn, the sodium concentration inside the cell is maintained by our sodium potassium ATPase pump. So this sodium glucose transporter indirectly utilizes ATP. So it is an example of secondary active transport. Understood? So here sodium dependent transport of ions and nutrients along their electrochemical gradient is secondary active transport. Here sodium is transported along the electrochemical gradient. Do you agree with me? Here the sodium concentration is less. So it is from higher sodium concentration to lower sodium concentration, the sodium is transported. Okay. But why does the sodium concentration less inside the cell? That is because it is taken out with the help of sodium potassium ATPase pump. Understood? So this is sodium glucose transporter, which is an example of secondary active transport. So the option A is right. And potassium hydrogen pump in gastric mucosa is an active transport mechanism. Yes, we have seen already it is an example of active transport, primary active transport. Osmosis is diffusion of water across the semi-permeable membrane from an area of high solute concentration to low solute concentration. Do you say yes for this? Osmosis we've been learning for such a long time. What is osmosis? It is the diffusion of water across the semi-permeable membrane. But what is the difference? It is the from an area of high solvent concentration or high water concentration. Okay, from higher water concentration to a lower water concentration. It is not solute. Okay, it is not solute, it is solvent, from higher solvent concentration to lower solvent concentration. That solvent is nothing but water here. Okay, so osmosis, this option is the wrong one. And hormones, digestive enzymes are transported outside the cell by exocytosis. So we've seen already what is exocytosis. Yes, we've seen already what is exocytosis. Okay, from what is exocytosis? It is externalization of any substances from inside the cell. So hormones are synthesized inside the cell, like for example, insulin inside the beta cells of pancreas. So this insulin has to be transported out so that it has effects outside the organ. So this insulin is externalized or exocytosed from the beta cells of pancreas. Also digestive enzymes digestive enzymes. So also digestive enzymes, they should be transported outside the cell by exocytosis. So this option is also right. So the answer for this question is option C. Iodide pump is an example of iodide pump. First understand where is iodide pump. It is present in the follicular cells. So here I have to transport iodine inside the cell, inside the thyroid cells. Okay. So it is done by sodium iodide symporter. Sodium iodide symporter. So if I have to maintain sodium concentration inside the cell, what will I be using? I will be using sodium potassium ATPase pump. Sodium potassium ATPase pump. So the same thing only. So for sodium iodide symporter to function, for sodium and iodine to enter inside the thyroid cell, the sodium concentration inside the cell has to be maintained. That is maintained by sodium potassium ATPase pump. So here sodium iodide symporter, this is nothing but the iodide pump that indirectly utilizes ATP. Okay, so it is an example of secondary active transport. Yes, it is an example of secondary active transport. Now GLUT transporters are example of facilitated diffusion. Yes, facilitated or diffusion is something but carrier mediated facilitated diffusion. So glucose transporter is the carrier or the transporter here through which glucose enters inside the cell, our cell. So there are different glucose transporters. How many different types of glucose transporters? GLUT1 and GLUT3 are present in RBCs, neuron, placenta. Okay, placenta. And GLUT2 is present in GLUT2. Where is it present? It is present in kidney, intestine, and liver. These three are involved in what? Regulation of glucose. Okay, these three organs are involved in regulation of glucose. And GLUT4 is insulin dependent glucose uptake by adipose tissue and skeletal muscle. These two are involved in insulin dependent glucose uptake. And there is something called GLUT5 which is helping in fructose transport, fructose uptake. Okay, it is mainly present in our sperms. So fructose uptake also in intestine. From the intestine also fructose is taken up. Okay, so here GLUT1, 2, 3, 4 and 5. There are many other glucose transporters, but importantly you have to know these 5. Marker enzyme for microsomes. Please learn the marker enzymes for all the organelles. It is very important. Okay. Microsomes, here they mention endoplasmic reticulum. Microsomes is nothing but endoplasmic reticulum. So what is where is catalase and peroxidase present? We've already seen it's present in peroxisomes. Yes, it's present in peroxisomes. So it's a marker enzyme of peroxisome. And acid phosphatase, which organelle has acidic pH inside? Which organelle has acidic pH inside? Lysosomes. So acid phosphatase is present in lysosome. So glucose 6 phosphatase is present in the endoplasmic reticulum. So the answer for this is glucose 6 phosphatase. Glucose 6 phosphatase. Yes, please learn the marker enzymes for all of the organelles. So what is the marker enzyme for mitochondria? For mitochondria, all enzymes of electron transport chain can be used as marker enzymes. So one is ATP synthase. Okay, and then for cytoplasm, what is the marker enzyme? Lactate dehydrogenase. Lactate dehydrogenase. Yes. So the coenzyme increases the affinity of apoenzyme to substrate, lowers the activation energy, increases the number of active site on the enzyme and accepts one of the products of the reaction. What do you think is the right answer? Coenzyme, what is the coenzyme? It is the non-protein part of the enzyme. Yes, non-protein part of the enzyme is of 3 types. One is prosthetic group. Okay, coenzyme and cofactor. Coenzyme and cofactor. They are actually involved in group transfers. Coenzymes are involved in group transfers. They transfer acyl group or they transfer oligosaccharide group. They transfer methyl group. So this is the function of a coenzyme. In certain textbooks, like in various other topics also, the all these three will be used interchangeably. Okay, so consider in one reaction a substrate uses NAD. Okay, NAD. So what happens from the substrate? Hydrogen is removed and it's accepted by NAD and it forms NADH, right? And then a product is formed. So here it actually accepts one of the products of the reaction. So the coenzyme is nothing but it accepts one of the products of the reaction. Also it involves in the group transfers, transfer of methyl group or acyl group or phosphate group. So these are involved in these are the functions of coenzymes. Okay, so does it increase the affinity of a coenzyme to the substrate? No, it does not increase the affinity. And does it lower the activation energy? No, enzyme actually lowers the activation energy, not the coenzyme. And increases the number of active site on the enzyme? No, this also cannot happen. So the answer is option D. And in competitive inhibitions, inhibitor has a structural similarity to substrate. Km is decreased, Vmax is decreased, reaction rate is independent of substrate concentration. So what do you think can be the answer? In competitive inhibitor, what happens in competitive inhibition? The inhibitor will be a substrate analog. Yes, so the inhibitor will compete with the substrate for the active site of the enzyme. The inhibitor will compete with the substrate for the active site of the enzyme. So this inhibitor should be a substrate analog. It should have structural similarity to the substrate. Only then it can bind with the active site of the enzyme. Yes. And also coming to Vmax and Km, what will happen to the Vmax and Km value? Km is nothing but substrate concentration at half maximal velocity. Yes, substrate concentration at the half maximal velocity. So here, if if the inhibitor is a substrate analog, if I try to increase the substrate concentration, then I can overcome the action of inhibitor. Yes, so if I increase the substrate concentration, I can overcome the action of inhibitor, and thus my Vmax can be unchanged. So if I have to bring back the control of velocity of the reaction, I can increase the substrate concentration so that I can overcome the action of the inhibitor over the enzyme. Otherwise, what happens in normal concentrations of substrate and inhibitor, the inhibitor can bind to the active site of the enzyme and it can decrease the reaction rate. But this is actually a reversible reaction. Being it a reversible reaction, why do I call it a reversible reaction? Because if I increase the substrate concentration, then my substrate's affinity towards the enzyme's affinity towards the substrate can be more. Okay, and more of the substrate can bind to the enzyme. Not the affinity cannot be more, but more of the substrate can bind to the enzyme and it can overcome the action of the inhibitor. Okay, so the increasing the substrate concentration, I can maintain the Vmax to be unchanged. Okay, so Km is decreased, it is wrong. Vmax is decreased, it's also wrong. So reaction rate is independent of substrate concentration. Yes. Is it independent? Reaction rate, Vmax is dependent on substrate. Only when you increase the substrate concentration in case of competitive inhibition, you can overcome the action of the inhibitor and thus the velocity of the reaction can be maintained. Okay, so reaction rate is independent of substrate concentration is also wrong. So the answer for this is inhibitor has structural similarity to the substrate. Okay, so this is about competitive inhibition. So how do you draw a line Weaver plot for a competitive inhibition? I'll draw substrate concentration, one by substrate concentration along x-axis and 1 by V along y-axis. So if this is the line without inhibitor, okay, this is the line without inhibitor. So wherein the y-intercept is 1 by V max and x-intercept will be minus 1 by Km. Here, my what happens to the Km value here? Km value, or what happens to Vmax? Vmax is unchanged. Yes, Vmax is unchanged. And what happens to the Km value? Km value is increased. So I'll have a point here. So this will be the line with inhibitor, with a competitive inhibitor. So this is the line Weaver-Burke plot for competitive inhibition. Yes. Now, digestive enzymes belong to the class of hydrolases. Okay, most of it will be what is the hydrolases? If you know the function of hydrolases, you'll be able to understand what are hydrolases. Hydrolases are the classification of enzymes which will break a linkage by adding water. Okay, break linkage by adding water. So what kind of bonds does it break? It breaks ester bonds, ether bonds, yes, peptide bonds and glycosidic bonds. Ester bonds, ether bonds, peptide bonds, glycosidic bonds are broken by adding water. Okay, so peptidases, we have peptidases which are digestive enzymes, right? So those will break bond by adding water. So those digestive enzymes come under the class of hydrolases. Those digestive enzymes come under the class of hydrolases. What are ligases? Ligases will be those enzymes that links molecules with the help of ATP. Okay, links molecules with the help of ATP. They are mostly carboxylases. Okay, so these usually carboxylases will need biotin, bicarbonate and ATP. Okay, so carboxylases come under the group of ligases. So example can be acetyl-CoA carboxylase or PRPP synthetase, because synthetase also comes under ligases. Understood? What are liases then? Liases, they break bond without adding water. They break bond without adding water. So decarboxylases, okay, decarboxylases and synthases come under this category. So with these three enzyme classifications are a bit confusing, but you have to understand. Hydrolases, they break bond by adding water. Okay. Hydrolases, they break bond by adding water. Liases, they break bond without adding water. Understood? And difference between ligases and liases is ligases are synthetases and carboxylases. Liases are decarboxylases and synthases. Yes? So, and they do not utilize ATP. Understood? So what is oxidoreductase? They catalyze oxidation and reduction reactions. That is oxidoreductase. Example can be any dehydrogenase, oxygenase, monooxygenase. These are all examples of oxidoreductase. And one of the following is an example of repression. So this is like enzyme regulation. Okay, enzyme regulation. What is ALA synthase by heme? ALA synthase by heme. Actually, ALA synthase is a rate limiting step of heme synthesis. Okay, so in heme synthesis, it starts with substrates succinyl-CoA and glycine. Okay, succinyl-CoA and glycine that For ALA synthase catalyzes this reaction forming ALA. And finally, what do we have? We have the heme. So heme is the product of this pathway. So increased heme. So when there is more of heme, actually we have to block this pathway. So what does heme do? Heme will try to heme will actually repress the genetic expression of the ALA synthase gene. So heme will repress or reduce the expression of the ALA synthase at the genetic level. Okay, so this is the mechanism of regulation of this heme synthesis. Okay, this is the regulation of this heme synthesis. Whereas succinate dehydrogenase by malonate, cytochrome by cyanide, these are all by inhibitory mechanisms. This is enzyme inhibition. Okay, enzyme inhibition. And glycogen phosphorylase by glucagon. What do you know about this? Phosphorylation mechanisms, covalent modification. Okay, this is regulation of enzyme activity by covalent modification. So what is this? Mainly enzymes are activated or deactivated by phosphorylation or dephosphorylation. Okay, so here I will tell you insulin. Let's take insulin and glucagon. Insulin and glucagon. So whichever pathways are stimulated by insulin, insulin will actually activate the enzymes by dephosphorylating them. Okay, insulin activates the enzyme by dephosphorylating them. Okay, insulin dephosphorylates enzymes and activate them. In other words, I will tell you, the enzymes are active in the dephosphorylated state. So what are the pathways which are stimulated by insulin? In carbohydrate metabolism, this is glycogen synthesis and glycolysis. They both are stimulated by insulin. In carbohydrate metabolism alone, I am taking as an example. So the enzymes involved in these pathways will be active in the dephosphorylated state because insulin dephosphorylates it and activates them. Okay, so these enzymes are active in the dephosphorylated state. Whereas glucagon, what does it do? Glucagon activates the enzyme by phosphorylating. So some enzymes are active in the phosphorylated state and some enzymes are activated in the dephosphorylated state. What are the pathways which are stimulated by glucagon? They are gluconeogenesis. Yes, gluconeogenesis or glycogenolysis. So these two pathways are stimulated by glucagon. The enzymes of these two pathways will be active in the phosphorylated state, and that phosphorylation is being induced by the glucagon. So some enzymes are activated in the phosphorylated state, some enzymes are active in the dephosphorylated state. So the hormones which will be regulating them also will be different. Okay, this is covalent modification. So glycogen phosphorylase is an enzyme of what? Glycogen phosphorylase is an enzyme of glycogenolysis, right? And that will be activated by glucagon by phosphorylating the enzyme. So this is a example of covalent modification, and B and C will be example of enzyme inhibition, and ALA synthase by heme, that is the example of repression mechanism. Okay, with the help of repression, it's regulating the enzyme activity. So heteropolysaccharide which does not contain uronic acid is. So you have to know about various mucopolysaccharides. Yes, you have to know about the various mucopolysaccharides. So here I'll write the heteropolysaccharide here, or the mucopolysaccharide here, and uronic acid, because a mucopolysaccharide is formed by repeating units of uronic acid and amino sugars. Repeating units of uronic acid and amino sugars. Okay. So first one, let's see, hyaluronic acid. Hyaluronic acid contains what? Glucuronic acid. Okay, uronic acid is glucuronic acid here, and amino sugar is N-acetyl glucosamine. N-acetyl glucosamine. Understood? Next is chondroitin sulfate. Chondroitin sulfate here, glucuronic acid. Uronic acid is the glucuronic acid here, and Amino sugar is N-acetyl galactosamine. Okay, galactosamine. Next, dermatan sulfate. Dermatan sulfate has what uronic acid? It has iduronic acid. It has iduronic acid. And amino sugar is N-acetyl galactosamine. N-acetyl galactosamine. And keratin sulfate, or let's first finish off heparin sulfate here. Heparin sulfate contains iduronic acid, okay, iduronic acid and N-acetyl glucosamine, N-acetyl glucosamine. So amino sugars, hyaluronic acid and heparin sulfate both have glucosamine, okay, both have glucosamine here. And glucuronic acid is present in hyaluronic acid and chondroitin sulfate. Iduronic acid is present in dermatan sulfate and heparin sulfate, okay. And lastly, keratin sulfate. Keratin sulfate contains, it does not contain uronic acid. Instead, what does it contain? It contains galactose. Okay, it contains galactose, sugar, and N-acetyl glucosamine. N-acetyl glucosamine. Yes, so keratin sulfate does not have uronic acid in them. Keratin sulfate does not contain uronic acid in them. Understood? So in this reaction, in this, what is the answer? Which does not contain uronic acid is keratin sulfate. Please don't forget, which does not contain uronic acid is keratin sulfate. Now, which is a non-reducing sugar? Non-reducing sugar is sucrose. Yes, sucrose. Why it is sucrose? Because the functional groups of glucose, sucrose is made of glucose and fructose. So the functional groups of glucose, it's an aldose, and fructose is a ketose. Both of them are connected in case of sucrose. So the functional groups are not free to reduce the Benedict's reagent. So this is a non-reducing sugar. Whereas maltose, lactose, isomaltose and glucose are all reducing sugars. Right, so see this here. This glucose and fructose, that is what is sucrose. Here the functional group of the glucose and functional group of fructose are connected. Okay, it is not free to reduce the Benedict's reagent. So glucose and galactose are consider this. This is the formula for glucose and galactose. So how do you number? You number them from top 1, 2, 3, 4, 5 and 6. There is 6 carbons, right? So here if you see along the 4th carbon, okay, along the 4th carbon the orientation is different. Yes, along the 4th carbon the orientation is different. In other carbons it is the same. So what is what is an epimer? What is an epimer? If the orientation of one of the carbon of the H and OH along one of the carbon atom other than the penultimate carbon atom is different, then it is an epimer. Okay, so only one carbon atom orientation should be different, only one, okay, then it is called epimer. So glucose and galactose will be called C4 epimers. Okay, glucose and galactose can be called C4 epimers. So other than which is the penultimate carbon atom, last before carbon atom is the penultimate carbon atom. So other than the penultimate carbon atom, if the orientation of H and OH along any one of the carbon atom is different, then that is a epimer. Okay, then that is a epimer. So glucose and galactose can be epimers. So the answer for this is epimer. Okay, and this is not a constituent of sucrose or they are not non-reducing sugars. They both are reducing sugars. And what is an anomer? What is the anomer? So consider here, in this is alpha and beta variety or the anomers. Okay, so in beta variety the OH group is above the plane of the molecule, and in alpha variety the OH group is below the plane of the molecule. So these two are anomers. Okay, alpha and beta variety, depending upon the orientation of OH group above or below the plane of the molecule. Okay, so these the anomers. So do you think glucose and galactose are anomers? No. For glucose itself there is alpha and beta anomers. And finally, reducing sugars are differentiated from non-reducing sugars by which test? The final question is, reducing sugars are differentiated from non-reducing sugars by. So you've got four choices: Smallish test, Benedict's test, iodine test and Selivanov's test. So think a Selivanov's test is for finding out keto group, right? So it will be positive in case of presence of fructose. Iodine is a test for polysaccharides. Yes, iodine is a test for polysaccharide. Smallish test is a general test for carbohydrates. Smallish test is a general test for carbohydrates. So whether carbohydrate is present or not only is detected by Smallish test. So what is Benedict's test now? Benedict's test is the test that differentiates reducing sugars from non-reducing sugars. Okay, so any substance that can reduce the Benedict's reagent are called reducing substances. Okay, so let's see the principle of Benedict's test. So Benedict's test, how will you perform this test? You'll add 5 ml of Benedict's reagent with 8 drops of the sample and heat it. Yes, when you heat it, you get the characteristic brick red precipitate. Characteristic brick red precipitate. So we have different series of colors like yellow color, green color, orange, and the brick red. So depending upon the concentration of glucose in the solution. Now let's see Benedict's test. What is the reagent? Reagent is copper sulfate, sodium carbonate and sodium citrate. So what are the reagents? Copper sulfate. Copper sulfate donates cupric ions needed for the reaction. Sodium carbonate, it provides the alkaline medium, and sodium citrate is a stabilizing agent, right? So consider glucose. Glucose or any reducing substances. What will happen? Glucose is converted to its enediol form. Glucose is converted to its enediol form. Enediol form is an unstable form, right? So in the alkaline medium, in the presence of alkaline medium, glucose is converted towards enediol form. Then the cupric ions will get reduced to cuprous ions, right? Cupric ions get reduced to cuprous ions, which in turn is converted to cuprous oxide. This cuprous oxide gives the color for the reaction. Now this enediol form will now get oxidized to sugar acid. Right, cupric ions get reduced to cuprous ions, which is converted to cuprous oxide. Cuprous oxide is responsible for the color of the reaction. Understood? So here, let's say we get different colors for the reaction depending upon the concentration of the glucose in the solution. Now, if it is green, yellow, red, orange red and brick red. So brick red indicates that the concentration is high. So that is why this test is called semi-quantitative test. Okay, that is why the test is called semi-quantitative test, because you can quantitate to some extent. It's a qualitative test. What is the difference between qualitative and quantitative? Qualitative is we can identify the presence or absence of a particular substance. Quantitative is we are able to quantitate or measure the amount of substance in the solution. Now here semi-quantitative in the sense, depending upon the color of the reaction, we are able to find out how much amount of glucose is present approximately in that particular test tube. Right, so it is a semi-quantitative test. So here the principle of Benedict's test is important, you all have to know. And reagent, what is the reagent constituents? So right, so what are the reducing substances? Reducing substances. Now we are talking about reducing sugars, right? So let's list out the reducing sugars. What are the reducing sugars? Glucose, yes, glucose, fructose, maltose, yes, maltose, lactose, these are all reducing sugars. Now, sucrose is a non-reducing sugar, we have seen already. And there are certain non-carbohydrate substances which reduce Benedict's reagent. Okay, so non-carbohydrate reducing substances are non-carbohydrate reducing they are not carbohydrates, but they reduce Benedict's reagent. So they also give positive Benedict's test. So here non-carbohydrate reducing substances will be vitamin C, uric acid, homogenticate, okay, and certain and drugs like your aspirin. So these are non-carbohydrate reducing substances that show positive Benedict's test. Yes. So one more question, which of the following enzymes contain molybdenum? So here you have to learn about metalloenzymes. Okay, you have to learn about metalloenzymes. Okay, so cytochrome oxidase. What is the enzyme metal present in cytochrome oxidase? It is copper. Okay, it is copper. Xanthine oxidase. Xanthine oxidase contains molybdenum. Xanthine oxidase contains molybdenum. And carbonic anhydrase, it is a zinc containing enzyme. Phosphoglucomutase, it is manganese containing enzyme. Okay, so these are the some of the metalloenzymes. All right. Thank you.